Hello students of Standard 9th. Today we will take up the next chapter of Science 2, Chapter 16, Heredity and Variation. Students, you already know that we inherit genetic characters from our parents. The branch of biology which studies the transfer of characteristics of organisms from one generation to the next, and genes in particular, is called as genetics. New progeny means offsprings, are formed through the process of reproduction. Except for a few minor differences, the offspring shows great similarities with the parents. The organisms produced by asexual reproduction show minor variations. However, the offsprings produced through sexual reproduction show comparatively greater variations. So if we compare some of the contrasting features like the facial characteristics, nose, lips, attachment of the earlobe or the hairline, we see that there are a lot of differences among individuals with respect to these features. However, if we compare parents and their offsprings, you will notice that there is a lot of similarity. So the transfer of characteristics from parents to offspring is called heredity. It is due to heredity that puppies are similar to dogs, squabs are similar to pigeons, and infants are similar to humans. Though there are many similarities between parents and their offsprings, there are some differences too. I am sure you all will agree that you are not exactly a carbon copy of your parents. These similarities and differences are all the effect of heredity. Let us study the mechanism of heredity. Information which is necessary for protein synthesis in the cell is stored in DNA. The segment of DNA which contains all the information for synthesis of a particular protein, it is called as a gene for that particular protein. It is necessary to know the relationship of these proteins with the characteristics of organisms. So let us move on to study more about chromosomes. Now, what exactly are chromosomes? The structure in the nucleus of the cells that carries the hereditary characteristics is called the chromosome. A chromosome is made up mainly of nucleic acids and proteins. During cell division, chromosomes can be very clearly seen under the compound microscope. Genes which contain the information about hereditary characteristics in a coded form are located on the chromosomes. Each species has got a particular or a specific number of chromosomes. Now each chromosome is made up of DNA and it appears dumbbell shaped midway during cell division. There is a constricted region on each chromosome and this is known as the primary constriction, or it is also called the centromere. Now this centromere divides the chromosome into two parts and each part is called as an arm. The centromere has got a particular position in each chromosome and depending upon this we can classify the chromosomes into 4 types. So let us study which are the 4 types of chromosomes. So types of chromosomes can be easily identified during cell division. Let us come to the first type of chromosome. This is the metacentric chromosome. Now in this type of chromosome, the centromere is exactly at the midpoint in this chromosome, and therefore this chromosome appears like the English letter V. The arms of this chromosome are equal in length. The second type of chromosome is the submetacentric chromosome. Now in this type of chromosome, centromere is somewhere near the midpoint, and therefore this chromosome looks like the English letter L. One arm is slightly shorter than the other. The third type is the acrocentric chromosome. In this type of chromosome, centromere is near one end of this chromosome, and therefore it appears like the English letter J. One arm is much smaller than the other. And finally, we have the fourth type, which is the telocentric chromosome. In this type of chromosome, the centromere is right at the end. So this makes the chromosome to look like the English letter I. This chromosome consists only of one arm. So here we have all the four types of chromosomes clearly visible. Metacentric, which looks like the letter V. Submetacentric, which looks like the letter L. Acrocentric looks like the letter J. And telocentric, which looks like the letter I. Now generally in somatic cells means the body cells, the chromosomes are in pairs. If the pair consists of similar chromosomes, similar in the sense they are same in shape and organization, then such type of chromosomes are known as homologous chromosomes. And if they are not similar, then they are called as heterologous chromosomes. In case of organisms which reproduce sexually, one of the chromosomal pairs is different from all the others. So the chromosomes of this different pair, they are called as the sex chromosomes or allosomes. And all the other remaining chromosomes are termed as the autosomes. Now we come to DNA. What exactly is DNA? Now the full form of DNA is deoxyribonucleic acid. Chromosomes are mainly made of DNA, and this acid was discovered by the Swiss biochemist Friedrich Miescher in 1869 when he was studying the white blood cells. Initially, this acid was reported to be only in the nucleus of the cells, and that is the reason why it was called as nucleic acid. But later on, it was realized that it is present in other parts of the cell too. Molecules of DNA are present in all organisms, right from viruses and bacteria to human beings. And these molecules control the functioning, growth and division, that is reproduction of the cell. And therefore, they are rightly called as master molecules. Now, the structure of the DNA molecule is the same in all organisms. In the year 1953, Watson and Crick produced a model of the DNA. And according to this model, two parallel threads of nucleotides are coiled around each other. And that is why this arrangement is termed as a double helix. You can compare the structure with a coiled and flexible ladder. Each strand in the molecule of the DNA is made up of many small molecules, and these molecules are called nucleotides. Now there are 4 types of nitrogenous bases: adenine, guanine, cytosine and thymine. Out of these 4, adenine and guanine are called as purines, while cytosine and thymine are termed as pyrimidines. In the structure of the nucleotide, a molecule of a nitrogenous base and phosphoric acid are each joined to a molecule of sugar. As there are 4 types of nitrogenous bases, nucleotides are also of 4 types. Nucleotides are arranged like a chain in a molecule of DNA. The 2 threads of the DNA molecule can be compared to the 2 rails of a ladder, and each rail is made up of alternately joined molecules of sugar and phosphoric acid. Each rung of the ladder is a pair of nitrogenous bases joined with hydrogen bonds. Adenine always pairs with thymine and cytosine always pairs with guanine. Now we come to the gene. Each chromosome is made up of a single DNA molecule. Segments of the DNA molecules are called genes. Due to variety in the sequence of nucleotides, different kinds of genes are formed, and these genes are arranged in a line. Now, genes play a vital role. They control the structure and the function of the cells and of the body. They are also responsible for the transmission of hereditary characters from parents to the offspring. That is why genes are said to be the functional units of heredity. And this is why many similarities are seen between parents and their offspring. All the information about protein synthesis is stored in the genes. Students, here you can see the structure of the DNA. With that, we come to the end of the first session of this lesson. You can go through this human genome project. Just read it, okay, for your information. So thank you until we meet in the next video.